Okay, everybody. Good morning, good afternoon, good evening, depending on where you are in the world. I hope you're all well, and that especially you remain so in these rather difficult times. I'm Mark Blaxter. I'm from the Wellcome Sanger Institute in the UK, where I'm head of the Tree of Life program, and it's my huge pleasure to welcome you on behalf of the Organising Committee of Biodiversity Genomics 2020 to this online meeting. So this meeting, just to explain, is an online avatar, if you like, of several other meetings which we had to cancel because of the coronavirus pandemic. So rather a meeting in person for the Earth Biogenome Project, for the Vertebrate Genomes Project, for the Global Invertebrate Genomics Alliance, and for the Darwin Tree of Life Project, we decided to move ourselves online. And it's been an amazing response. There's about 3,000 people have registered from over 70 countries. And we hope to see some or all of you at various different points in this conference. So just say what the meeting's for. We're gathering to celebrate the diversity of all the species and systems that we work on. And whether this is from protists, animals, fungi, plants, and whether you're interested in sample acquisition in the legal and ethical side of this, through sequencing, through assembly, through annotation, and through exploitation of these genome sequences for all sorts of interesting biology that you want to do. We hope you'll find something of value here. I think we're also celebrating another thing in this meeting. That's the diversity of people who are involved in this great escapade of sequencing life. And in this year, we have a special focus in the context of Black Lives Matter, and also in the context of the United Nations Summit on Biodiversity, which has just happened. And we really want to foreground the development of our community during this meeting. We want to promote diversity and inclusion in our community, and also pay special attention to lower and middle income countries and younger scientists who are the ones who are going to take this great project forward. So please, these discussions will happen in fora as well as in other parts of the meeting and informally, but please do join these forums and hear what people have to say and think about how we can make our community, how we can grow our community, and how we can assure that the future of our community is both inclusive and open. As part of that, we do have a meeting code of conduct. It's on the AV portal landing page. Do have a look at it. The code of conduct is relatively standard for meetings of this kind. Because the meeting is open and being broadcast, and we really don't have much to say about social media, because this is who we are. But the basic content of the code of conduct is to be kind and be inclusive in all your interactions with colleagues over the next 5 days. As part of our online contact, we also have a Slack channel. Please do join that and see all the different rooms in the Slack channel that you can meet friends and new colleagues and post information. And we also have meeting chat rooms. So these will be open 24/7, so you can arrange to meet people there anytime. And please do use them to meet with your colleagues, to view the posters, that's where the poster rooms are, and to experience our diversity. So the meeting's been made possible through funding from some generous sponsors. And that includes Dovetail, Pacific Biosciences, and Oxford Nanopore Technologies. These sponsors are at the meeting and they have dedicated chat rooms, so please do go and catch up with them in their chat rooms, and thank them very much for their sponsorship of the meeting. The meeting is also being sponsored by the Wellcome Sanger Institute through the Tree of Life program. So welcome, welcome. Please do enjoy the meeting. As I said, there's nearly 3,000 people registered, over 70 countries, and we're humbled and honored to greet you all. So in normal times, this meeting would have happened in person on the campus of the Wellcome Sanger Institute in Hingston, the UK. And normally there would have been a welcome from the director of the Sanger Institute, Mike Stratt. So Mike has recorded a welcome for you to welcome you all to the meeting. Just for those of you who don't know Mike, Professor Sir Mike Stratton is director of the Wellcome Sanger Institute. He's been at the Sanger Institute since 2000, where he's been driving large-scale analyses of human genomes, especially in the context of cancer and somatic mutation. So he's an FRS, a Fellow of the Royal Society. He's many other honours, and it gives me great pleasure to now introduce Mike Stratton to give his welcome to you all to this meeting.
Hello, I am Mike Stratton, Director of the Wellcome Sanger Institute in the UK. I'm very pleased to welcome you all to Biodiversity Genomics 2020. Many of you will know the Sanger Institute as one of the centres that generated the reference human genome, and we've been sequencing many more human genomes since that momentous first assembly 20 years ago. What may be less well known is Sanger's involvement in what could be called biodiversity-focused genomics. We have been involved in the sequencing of model organism genomes, including many vertebrates that help us better understand the human, and the standard laboratory models, such as the nematode C. elegans, which was in fact our, and indeed the world's, first animal genome sequence. In the area of human infectious disease, we have also sequenced many bacterial pathogens, protozoa, nematode and flatworm parasites, and also the vectors that transmit these debilitating diseases. From our viewpoint, these diverse genomes have not only delivered to our goals of improving human health through genomics, but have also opened our eyes to the variety of ways that genomes are constructed and the ways in which having a genome sequence can change the way biology is done. We have therefore now committed as an institution to deliver genome sequences across the tree of life. We're doing this on the basis that in the midst of the environmental crisis that threatens the survival of both human society and the ecosystems on which we depend, we will use our quarter century of experience in delivering the highest possible quality of sequence and our ability to generate data at scale to deliver to the world high quality genomes that will be freely accessible by all. So it is a personal pleasure to be present at the start of a new revolution in biology, the sequencing of all life, and to be present at this meeting that celebrates the inclusivity and diversity of the growing scientific community that is building to meet the goal of sequencing all life for the future of life. I hope you have an exciting, enjoyable, and inspiring meeting.
Thank you very much, Mike. So now on to the first formal presentation of our meeting. This presentation is from Professor Harris Lewin. So Harris Lewin's the Robert and Rosabelle Osborne Chair of Evolution and Ecology at the University of California, Davis. He joined UC Davis in 2011, and he's been Vice Chancellor for Research. Previous to that, he was at the University of Illinois for 25 years, where he, for example, amongst many other things, helped found and establish the Keck Center for Comparative and Functional Genomics. So Harris has been working in our area for most of his career, and for many of us for as long as we've been alive. Harris's science focuses on the comparative genomics of mammals, on the evolution of species, on adaptations, and especially of cancer. And more recently has been involved in looking at mammalian genomes with respect to coronavirus. So the reason Harris is talking to us today is because in 2017, along with others, he co-founded the Earth Biogenome Project. And he is chair of the EBP working group and continues to inspire us all. So Harris's huge enthusiasm and vast vision is one of the things that has made this project possible. I'm really honored to now introduce Harris to open the meeting with, if you like, a state of the nation or a state of the world address as to how we're doing with the Earth Biogenome Project. Harris.
Thank you, Dr. Stratton, and a big welcome to more than 2,500 people who have registered for this historic virtual conference on biodiversity genomics. I want to begin by thanking the Wellcome Sanger Institute for its leadership role in organizing this conference, and the Wellcome Trust for providing the financial support for making the conference free to everyone, anywhere in the world. I'd also like to acknowledge support from our corporate sponsors and my home institution, the University of California, Davis, for providing resources for housing and supporting the Secretariat of the Earth Biogenome Project. Big thanks also to our conference chair, Mark Blaxter, and my co-chairs, Erich Jarvis and Joe Lopez, for their outstanding efforts to bring this conference online under such challenging global circumstances. Over the next 4 days, this conference, you will hear from many scientists at the forefront of the new and exciting area of biodiversity genomics. This conference has its roots back in 2009, when Steve O'Brien, Oliver Ryder, and David Hausler organized the first meeting of the G10K Consortium, an effort to sequence the genomes of 10,000 vertebrate species. It was an idea that was ahead of its time. I was fortunate to be at that first meeting, which inspired me and others in many ways. G10K spawned other efforts, including the Global Invertebrate Genomes Alliance, chaired by conference co-chair Joe Lopez, held its first meeting in 2013, and the Vertebrate Genomes Project, a sub-project of G10K, chaired by conference co-chair Erich Jarvis, which held its first meeting in 2016. In 2015, the Proto-Earth Biogenome Project held its first meeting at the Smithsonian Institution, hosted by EBP co-chair John Kress, to explore the rationale and feasibility of sequencing all of life. 2 years later, in 2017, we organized the first biodiversity genomics meeting at the Smithsonian, where we publicly rolled out the concept for the Earth Biogenome Project. The concept gained many enthusiasts and led to the publication of our perspective piece published in 2018, and subsequently the official launch of the project in London later that year. In August of 2019, Erich Jarvis and I co-chaired the first joint meeting of the G10K Consortium, the Vertebrate Genomes Project, and the Earth Biogenome Project at Rockefeller University in New York City, a meeting that was an important milestone that set the stage for the rapid progress and expansion of scientific interest in large-scale biodiversity genomic projects. And today, we begin Biodiversity Genomics 2020 with more than 2,500 registrants to learn about global progress in meeting the goals of the Earth Biogenome Project. We've really come a long way these past few years. My comments today will inform the current status of the Earth Biogenome Project and related projects, and where we are headed in the coming years. The world is at a critical inflection point. Climate change, deforestation, pollution, and other human activities are causing a rapid extinction of species, which is threatening Earth's vital ecosystems. The scientific evidence tells us that a 6 mass extinction of life on Earth is already upon us. Extinction rates of plants and animals have increased hundreds to possibly 1,000-fold above background rates. The Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, or IPBES, estimates that 1 million plant and animal species may be at risk of extinction by the end of the century. The problem now is receiving worldwide attention and concern. Last week, the UN held a summit on biodiversity, where the Aichi goals that were set back in 2010 for biodiversity conservation were reviewed and discussed. The sad and frightening findings presented in the Global Biodiversity Outlook 5 report was that none of the 20 goals set in 2010 have been met by the nations of the world. For example, 60% of the world's coral reefs are under threat from warming of the oceans, ocean acidification, and pollution. As a result of the worsening conditions, today, about 25% of the Great Barrier Reef has now completely bleached. Degradation of other ocean, freshwater, forest, and desert ecosystems has continued, and we are now faced with the possibility of massive ecosystem collapse on a global scale. Such a collapse will have enormous impact not only on biodiversity, but also on human populations and possibly global stability, and might ultimately affect our own survival as a species. Those services provided to us by nature that generate food, clean air, and water, that regulate critical environmental processes such as the biogeochemical cycle, and are the basis for deep cultural and aesthetic ties of humans to the natural world. Biodiversity is also the foundation for a new and rapidly growing bioeconomy that exceeds $500 billion each year in the US and the EU alone. If biodiversity disappears, so too will disappear the potential for the new bioeconomy to fuel what has been identified by the World Economic Forum as a key to the fourth industrial revolution. In response to these challenges, after nearly 3 years of planning, a group of scientists from around the world proposed to sequence all known eukaryotic life on Earth. This birthed the Earth Biogenome Project. So what is the Earth Biogenome Project? The Earth Biogenome Project is a confederated international network of networks that has the common goal of sequencing the genomes of all 1.5 million known species of eukaryotes in 10 years. So what has the EBP become today? The main role of the EBP organization is to develop and promote standards for whole genome sequencing, dissemination of best practices, and coordination of sequencing, analysis, annotation, and training activities. Also, the EBP leads the development of ethical standards for the project participation, for data sharing, and access and benefit sharing of intellectual property derived from whole genome sequencing. Finally, the EBP, its website, and social media arms are the central points for communications about the project. The EBP network, global network of networks, functions to support the 3 proposed phases of the Earth Biogenome Project. Phase 1, an annotated reference genome for 1 representative species of each taxonomic family of eukaryotes in 3 years, about 9,300 species in all. Phase 2, a reference quality genome for 1 representative of each eukaryotic genus in years 4 through 7, about 180,000 species in all. And phase 3, a reference genome for 1 representative of each eukaryotic species in years 7 through 10, an additional 1.32 million species. The EBP member institutions are committed to signing the Memorandum of Understanding to open access and compliance with the Convention on Biodiversity and the Nagoya Protocol on Access and Benefit Sharing. The EBP organization has a secretariat that's located at UC Davis and is operating under an interim governance structure. The main activities related to standards development are conducted under the International Science Committee that is headed by Stephen Richards here at UC Davis. There are 5 subcommittees: sample collection and processing, chaired by Mara Lonacek; sequencing and assembly, chaired by Richard Durbin; annotation, chaired by Paul Flisek; IT and informatics, chaired by Xiaofeng Wei; and data analysis, chaired by Shustin Lindblad-Coe. Major progress has been made by these committees during the past year, and following the meeting, all the committees' reports will be available on the EBP website. The EBP also has a committee on ethical, legal, and social issues, chaired by Hank Greeley from Stanford. Hank, Melissa Goldstein, and co-chair Jake Shurko have put together a stellar committee that will make recommendations on legal obligations relating to the Nagoya Protocol and access and benefit sharing, ethical considerations relating to collection of samples, societal concerns, and national and global security issues, and collaboration standards on sample information, data, intellectual property, authorship of papers, and publication guidelines. The LC session will be held on Tuesday, October 6th, and you are all encouraged to attend and participate. The Earth Biogenome Project now has 33 member institutions in 18 countries that have executed the Memorandum of Understanding, with 9 new institutions in 4 countries joining in the past year. A notable deficiency in an area of the world that is rich in biodiversity is the African continent. Progress is rapidly being made in developing the infrastructure for EBP in Africa, thanks to the efforts of Thank God Ebenezer. You are all invited to attend a session on Tuesday to learn about EBP-related activities in Africa. In addition to the institutional members, there are also 27 affiliated projects that are focused on national and regional efforts in specific taxonomic groups of organisms. I will present to you now brief summaries of 4 of the most well-funded and successful projects to date. You will hear more about these projects and others during the course of the conference. First, the Tree of Life program at the Wellcome Sanger Institute, led by Mark Blaxter, our conference chairperson. The Darwin Tree of Life project has a goal of sequencing all 60,000 known species in the UK and Ireland, while working collaboratively on several global efforts, including the Aquatic Cyst Symbiosis Genomics project that is funded by the Gordon and Betty Moore Foundation. The British and Irish biota include 4,061 families, or 43% of the known total of taxonomic families. Sequencing a representative species of all these families will contribute significantly to the phase 1 goals of the Earth Biogenome Project. Rapid progress in sequencing reference genomes has been made at the Wellcome Sanger Institute during the past year. Fresh, high-quality samples for 685 species have been received. 170 are currently being sequenced, and 69 have been sequenced to the proposed EBP standard. The Sanger has current output of 24 reference genomes, reference quality genomes per week, and is planning to increase output to 40 genomes per week by spring of 2021. Another project is the Vertebrate Genomes Project, a global consortium led by conference co-chair Erich Jarvis at Rockefeller University. The VGP aims to sequence 70,000 species of vertebrates. In its proof of principle phase, hoped to be completed in 2021, the VGP has selected representatives of all eukaryotic orders among the 5 vertebrate classes for sequencing. In the past year, the consortium released 101 reference quality vertebrate genome assemblies to the world. This is a significant milestone, far surpassing all previous efforts. The 10KP project is a global consortium led by Xin Liu of BGI in Shenzhen, China. The project aims to sequence 10,000 of 385,000 known species of plants, as well as a large number of single-cell eukaryotes. So far, the project has completed 225 genomes from the major taxonomic groups of plants, including many involved in food production, medicinal plants, and plants with unique adaptations. And the final project I'll talk about is the California Conservation Genomics Program that is directed by Brad Schaefer at the University of California, Los Angeles. The CCGP will use genomics to create management plans for threatened species in the state of California. 200 threatened species will be studied that are distributed across the entire state of California and all of its ecological zones. The project will involve 70 PIs at all 10 UC campuses. Sequencing and assembly is being done at UC Davis, UC Berkeley, and UC Santa Cruz. For this project, 150 reference genomes will be produced to meet EBP goals. Also, resequencing of about 100 individuals from each threatened species will be drawn from different populations around the state to identify genetic polymorphisms that can be used to understand population history and existing genetic diversity. These data will be used to develop management plans, species conservation. The area of conservation genomics is rapidly taking on increasing importance. I would like to point you to the plenary lecture on Thursday by Carolyn Hogue, who will discuss new large-scale conservation projects in Australia. So what is the status of global biodiversity sequencing? Of the estimated 12 to 15 million eukaryotic species on Earth, including 1.1 million species of plants and animals, there are about 1.5 million species that have been described by humans. Of those, and please note the log scale here, there's a representative species from 81% of the 70 eukaryotic phyla, 64% of the classes, 39% of the orders, 15% of the families, 2% of the genera, and less than 0.4% of all species, up only 0.1% from the previous year. Thus, our current understanding of the diversity and evolution of life on Earth is extremely limited by the lack of availability of genome sequences for most of life on Earth and the quality of the genomes that have been produced thus far. This slide, created by Stephen Richards, shows the distribution of assembly quality in terms of contig N50 and scaffold N50. The vast majority of the 5,698 unique species that have been sequenced fall into short read draft category, with contig N50 less than 100 kilobases and scaffold N50 less than 10 megabases. A small number of those draft assemblies have achieved greater contiguity to chromosome scale using scaffolding methods such as Hi-C. Recently, there's been a burst of chromosome scale assemblies produced using long reads and Hi-C, with most of these coming from the VGP, the Vertebrate Genomes Project, and other EBP-affiliated projects. Stephen Richards took a deeper dive into these data to reveal the chronology of assembly quality. The number of whole genome assemblies at all levels of assembly quality has nearly doubled in the past 2 years, from about 3,000 to the 5,698 number that I mentioned before. Most of these are draft quality, but the impressive statistic is that the number of reference quality chromosome scale assemblies has nearly doubled in just 1 year, from about 210 to 410. This is extraordinary growth that will be necessary for the EBP to meet its targets. For example, if we look at the EBP phase 1 goal of 9,300 families, although the number of reference quality genomes at the family level has doubled in the last year, we will have to produce about 40 times more reference genomes over a 3-year period, nearly 3,000 per year on average, to meet the EBP target. And that brings us to the technical challenges ahead, of which there are many. These include sourcing and vouchering thousands of specimens, high molecular weight DNA and RNA isolation at scale, sequencing capacity and throughput, assembly and curation at scale, annotation at scale, managing the large data flows, whole genome alignments at scale, and comparative genomic analysis and data visualization at scale. As mentioned before, there are EBP subcommittees dealing with all of these issues, and the reports will soon be posted on the EBP website. I'd like to return to the issue of sequencing capacity and throughput, as it relates directly to meeting the EBP targets. To meet the EBP phase 1 goals, we will need to produce 9 genomes per day, 365 days a year. Is this feasible? Well, Sanger alone will be producing 40 reference quality genomes per week in 2021, corresponding to 6 genomes per day. It is clear then that with the current technology and the number of labs coming online this year, many with expansion plans, we should be able to meet the goal of 9 genomes per day globally without any difficulty. This is very exciting news. And the other very exciting news, if my phone would stop ringing, is that the cost of producing a reference genome is only 1/3 of the cost of what it was 2 years ago. The project will save $186 million in phase 1 due to these cost reductions, bringing the total cost of phase 1 to about $414 million, down from $600 million estimated in 2018. To achieve the outputs for phase 1 and phase 2, dramatic increases in efficiency will be required. To sequence representatives of 180,000 genera in 4 years, the output of genomes will need to increase to 123 per day, or 14-fold. And for phase 3, another 10-fold above that to complete the project in 10 years. We're actually optimistic that the technology will improve and costs will be reduced so that reference quality genomes can be produced for all species for under $1,000 per genome. I'll remind you that 2 years ago, the cost, accuracy, and contiguity of assemblies we are achieving today with long reads was simply not on the radar. We have about 3 years to get there, and I think we will. Of course, one of the greatest challenges is finding the funding for all of this, estimated at $4.1 billion globally back in 2018, which is actually less than the original cost of sequencing the Human Genome Project, the Human Genome rather, in today's dollars. Sequencing that is still ongoing, I might remind you. On Friday, there will be a funders forum that I invite you all to attend. The forum will explore current funding for EBP, funders' perspectives on EBP and funding priorities, and prospects for a global funding consortium. Again, please join us. It should be a very interesting and informative conversation. Now, there are 3 big questions that we face in executing the Earth Biogenome Project. First, why now? It's because we must, and we know it will be hard. But the clock is ticking, and we may be running out of time. Much of life on Earth, on which we depend, is in great danger. And as David Attenborough said last week, extinction is forever, so our action must be immediate. Second, why sequence everything? Why not just eukaryotic families, or stop at the eukaryotic genera? The simple answer is this: you cannot predict which organisms will lead to the next breakthrough in our understanding of life, or the production of valuable new sources of pharmaceuticals, of food, or energy. No one could have predicted that the little tardigrades, the water bears, would tell us how DNA could be protected against extreme environmental conditions using the D subprotein, or that jellyfish might hold the key to tissue regeneration through the Wnt-beta-catenin pathway, or that comparative genomics of the ACE2 receptor might lead us to the intermediate host for the SARS-CoV-2 virus. Of course, that's just a small sample of the many revolutionary advances in knowledge that biodiversity genomics has brought us. To preserve life on Earth, we must not just learn how to save individual species, but also to preserve and restore entire ecosystems. Although the technology to do this is not yet here, having complete genome sequences for all organisms in an ecosystem is necessary for fully understanding how the ecosystems function and ultimately how they might be saved or reconstituted. And finally, the third question, why us? First, because we can. We have the technology, we have the expertise, and we have the passion. But most importantly, as scientists and citizens, we have the moral responsibility to do everything we can to save our planet for future generations. According to the Judaic concept, we should all practice tikkun olam. We should all work to repair the world before it is too late. I want to thank you all very much for your attention and enjoy the conference.
Thank you very much, Harris. It's really difficult to know how to applaud in this virtual way, but if you can imagine deafening applause, that would be good. I'm sure there is. So some questions have been posted on the plenary channel. I'm going to ask them to you. Some of them are anonymous, some of them have got people's names attached. So Philip Schiffer wondered what you thought about the rate of extinction of non-vertebrate, so invertebrate taxa. So are we underestimating that? Are they more important than the extinctions we are spotting in terms of the larger taxa?
Thanks for that question, Phil. I think the extinction rates in invertebrates is largely underestimated. It's really unknown. Most of the estimates that we have are for plants and animals by International Union of Conservation of Nature, things that we could readily see and count. Underestimating the extinction rates in invertebrates is also a very dangerous thing, because we know that these invertebrates are a critical part of the food chain. And if the invertebrates also begin to go extinct, it could have a dramatic effect on the entire food chain, especially in the oceans.
Yeah, it's a sobering thought when you think about recent data on the population decline in many species of insects in many developed nations, especially.
Yes.
Might mean for the stability of our ecosystems, especially agri-ecosystems.
Didn't mention insects, and that's very well documented. The case in Puerto Rico is a good case in point of what happens when insect populations collapse.
So somebody just tapped in deafening applause, Harris, so there you go. Another question is, how do we sample from endangered species? How many samples do we need? How many specimens do we need to sample? And what's the ethics of sampling from truly endangered species?
Yeah, that's a great question, and one in which our Ethics, Legal, and Social Committee is giving quite a bit of attention to. It's often not easy, it's not even legal to kill an endangered species to obtain a sample. So, when you're lucky, you might be able to obtain a sample that's been, that has been recently died, or obtaining a sample from an individual of an endangered species. And the zoos have been incredibly helpful in this regard. I might add that there's a big effort underway by people like Oliver Ryder at the San Diego Zoo to establish cell lines from as many threatened and endangered species as possible. And of course, cell lines give us a perpetual source of DNA and RNA for future studies, genomic studies of species that are threatened and endangered and may even become extinct.
Yeah, and there is a session on genomic resources. So resources.
Yes, there is.
Yeah.
And conservation.
Conservation later in this. So a question which has been liked by lots of people. I'll just read it out. There seems to be a lot of talking between nations and organizations, but what action has already started to improve biodiversity, and what are the barriers to action? So I guess this is what action has already started to happen because of genomics. We can't answer for the entire world's efforts in biodiversity, but how is genomics helping already? And what do you think are the barriers to letting genomics assist more effectively?
Yeah, most of the conservation problems, tend to be national or regional, and many of the efforts, such as with the Tasmanian devil in Australia, have taken national funding and national funding priorities, and those efforts tend to be integrated, multidisciplinary efforts in which genomics has played a very important role in characterizing population, genetic diversity, population history, and being able to identify genetically diverse populations for conservation and restoration efforts. Other efforts, such as the large megafauna, like northern white rhino, those tend to be really global efforts involving scientists from all over the world, with international funding sources that are aimed at conservation of specific species. And there have been notable successes in this area. And I think these successes, we've learned so much from efforts like the northern white rhino, the California condor, the Tasmanian devil, and others, that this is sort of breaking the ground for understanding the requirements for this moving forward. But as I mentioned in my talk, saving a species so that it exists in a zoo is is a noble objective. Saving species so that they can exist in their natural habitats in the wild requires saving and preserving entire ecosystems. And I think this is really important and part of the Earth Biogenomes efforts. Mark, I know you'll talk about Wyndham Woods in your talk, Wytham, sorry, that preserving, understanding the entire ecosystems is really from the bottom up is I think prerequisite to conserving it. And for those of you who are interested in it, David Attenborough had a wonderful premiere on Netflix last night for his terrific new series, and I urge all of the attendees at the conference to spend an hour and 20 minutes to watch David Attenborough's new documentary, which deals specifically with this issue.
So I'm going to praise
Attenborough.
David Attenborough, yeah. I'm going to praise a couple of questions that have come in, which are to do with where the division comes between doing whole genome sequencing in terms of enhancing conservation efforts, and where just a handful or many genomic markers might do the same job or might be more effective. I wonder if you can talk about the balance you feel between where doing a whole genome sequencing of an organism is obviously the EBP's goal, but maybe for conservation genomics, all that is needed, and need is an interesting question, all that is needed is a few markers so that pedigrees can be traced, so that biogeography can be tracked. So what do you think is the special role that genomics, whole genomes, play in this process?
I think there's a broad consensus in the field now that a reference genome is extremely valuable for conservation genomics program. The California Conservation Genomics Program debated this issue for months and firmly came down on the side of having a reference genome, at least at the genus level, for all species that are going to be targeted in this program. And that, I think, the reasons for that were very clear. If you really need to get to the genetic level to understand the genes that are important for adaptation, you have to have a whole genome sequence. And so the value I guess there's but there's value across the scientific spectrum in having the reference genome sequence when you're trying to do conservation at the species level. I think Carolyn will cover this very well in her talk because they have a big new conservation genomics program for threatened species in Australia, and there's an entire session on conservation genomics later in the meeting that will also be specifically dealing with this effort, and I urge the listener to tune in.
Yeah. So Marco has asked, what about the richest ecosystems in the world, such as Brazil and the African countries? Are they a focus for funding of genomic studies? How are we going to fund sequencing of these very, very rich and biodiverse ecosystems?
Yes, particularly in the areas where there are tropical rainforests, which are among the most biodiverse areas of the world. I will point you to another talk that we're going to have, a plenary talk. The situation in Colombia, EBP Colombia, I think is a really instructive example on how this is going to happen and unfold. There are parallel efforts in Brazil being funded by FAPESP, but the Colombian situation is really far along. Federica de Palma, Silvia Repestro, who will be talking about this as well. The Ministry of Science in Colombia has made the EBP, or their Biogenomics Colombia program, as a flagship scientific program, national scientific program that is getting underway this year. Most countries that we have been talking to that are participating in the Earth Biogenome Project are extremely enthusiastic about building scientific capacity and sequencing, or at least participating in the sequencing and analysis of genomes from species that are endemic to their own countries. And I think this is the way it's going to unfold, just as it did in the United Kingdom with the first announcement for the Darwin Tree of Life to sequence all 60,000 species in Britain and Ireland. This model is being replicated in many countries throughout the world, and I think that's the model that we are looking toward. Now, there are many countries in the world that will not have the infrastructure or the scientific expertise to do that. And I think one of the beautiful aspects of, for example, the Darwin Tree of Life program is its internationalization, its willingness to train scientists from all over the world in sequencing and assembly. And I think this connection between the developing African projects and the Sanger and the Wellcome Trust is going to be a very important model for other regions of the world. So I'm very hopeful that in even regions where obtaining, funding, direct funding from national governments may be difficult or impossible, that the wealthier developed world will be able to step in and participate in capacity building and then in sequencing endemic organisms to those areas of the world, countries and areas of the world.
Thank you, Harris. Just to say, the most liked question on the board now is thunderous applause by several fold. And we've got about 30 seconds, which is probably not enough. But one of the questions was asked very early on, which is quite popular, is what are we going to do after we complete phase 3? I love the optimism. What are we going to do when we complete phase 3? Well,
hopefully we'll still be here to see that, Mark, all of us. And, it's something that, it would just seem like a pipe dream to us, but is really happening fast. And I have no doubt that this is going to happen. And so when we do hit the number of 1.5 million, we must remember that there's probably 8 million more behind that have not been recognized by science. And so for all of you young people out there, there's going to be lots to do for a long time. And so get on your prospecting clothes, go out, collect, identify new organisms and species. And I think another 8 million will keep the world busy for some time.
Okay. Thank you very much, Harris, and
You're welcome.
also Mike. With that, we'll close this first session of Biodiversity Genomics 2020. Thank you all for logging in. There's now going to be a 10-minute break in the presentations for the meeting. The chat room is open. Chat rooms are open. Do go and visit Spatial Chat Rooms, visit our sponsors, and we will see you back here on this channel in 10 minutes to listen to our first plenary from Erich Jarvis of the Vertebrate Genomes Project. Thank you very much, Harris.
Welcome back, everyone. Our next plenary speaker is Professor Erich Jarvis from Rockefeller University and a scientific investigator with the Howard Hughes Medical Institute. As you'll soon find out from Erich's talk, Erich's a pioneer in the study of brain pathways in songbirds as a model to give new insights into how these animals produce and use vocal communication. Erich has been a dynamic leader in the genomics community. He's been a strong advocate for diversity in science. Erich is currently serving as chair of the G10K Consortium and the Vertebrate Genomes Project and is a co-chair of this conference. We have all come to appreciate Erich's fearlessness, his determination, and his absolute commitment to the Vertebrate Genomes Project and the production of high-quality genomes at scale. I now give you Professor Erich Jarvis.
So I want to thank the organizers for suggesting I give this keynote. I'm actually one of the organizers, but it was the others who suggested for this plenary, that is. And this title, I'm going to blame it on Mark Blaxter. He is the one who suggested it to me. And I can see how the work that I've been doing personally in my lab and in collaboration with the Vertebrate Genomes Project, the EBP, and others relates to it. Because my interest is in understanding how genes and the genomes they're in contribute to complex traits, and particularly complex behaviors, like vocal learning, the ability to produce learned sounds, as the species you can see in the background here, including our own human species, with Jennifer Lopez representing us. And the trek on understanding the genetics of these complex traits has led me to become more involved in genomics because of issues of genome quality. And what I'm going to talk about today is some of the challenges we've had on trying to study the genetics of complex trait with error-prone genomes, what we've done about them to try to develop platinum quality genomes, and the impact that has on large-scale genome projects. So the first topic here, the complex trait that I actually study the most is spoken language, its ability to imitate sounds and pass on culturally novel sounds from one generation to another. This ability, parts of it or components of it are found in other species like auditory learning, vocal usage learning, and there's some aspects of it that is unique to humans or to a few groups of species like vocal production learning, which is found in 5 groups of mammals: humans, dolphins, bats, elephants, and seals, and 5 groups of birds: parrots, songbirds, and hummingbirds. All of these species have close relatives like non-human primates to humans that don't learn how to imitate. And so all of them are thought to have evolved this trait independently of a common ancestor. Yet another trait we depend upon for spoken language is auditory learning, and this is more ubiquitous. Like your dog can understand the word sit, or siente se in Spanish, osuari in Japanese, roll over, get the ball, and so on. But a dog can't say, okay, you got it, I will sit, or tell you to sit yourself. Instead, a dog goes woof. And a dog can learn how to produce its woof sounds in different contexts, which we call vocal usage learning, but it doesn't actually learn how to imitate the sounds like these other species do. Now, not all vocal learners are as good as humans, but some are pretty far advanced, like parrots here. And this is a parakeet. And here's an example of what an animal named Disco could do after being raised with humans for 4 years.
Never shake a baby bird. That would surely be absurd.
Never shake a baby bird. That would surely be absurd. I am not a crook. My name is Disco. Again, meow, meow. Give me a kiss. Hey, kid, has cheeseburger. Are you a little stork or a stormtrooper? Disco, I used to be a spy. And a parakeet. Sure, I'm a doctor, not a parakeet. I did. I'm a doctor.
So this is amazing. You have this parakeet with a brain much smaller than ours, imitating all these human words, can recombine them into new meanings. A lot of times it's random, but in many cases, it's also appropriate for the situation, like when a dog shows up here, he goes meow, kind of gets the category correct, but the actual individual type of animal wrong. So this is remarkable. Yet our closest relatives, some people get excited when they're doing something that might approach what we find for spoken language, but they're still not as good as these other animals. And here is the best we can get from some of our closest relatives. A great ape, a Coco, raised with humans for 39 years.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
That is excellent. So here what's going on is Coco has good auditory learning, understands all these human words that are being asked of her, and is able to voluntarily do some type of sound production. In this case, a cough, but it's mostly a voiceless type of sound. Coco has difficulty modulating the sounds going through the larynx to produce learned vocalizations, much like we do and these parrots do for their syrinx. And so scientists such as myself and many others have been looking at the brains of all these various different species with the ability to imitate, like songbirds in humans, and those that don't, like the chickens and so on. And what we have found is that all of these species have in blue this auditory pathway, which I think is inherited from a common ancestor of vertebrates, but only the vocal learning species have these four brain regions in yellow and red that are responsible for producing the learned sounds and for imitating them, that you can't find in the other species, or you find them in only rudimentary fashions. So we've been asking, and I've been asking, what is the genetics of this trait? How did it evolve? Did it evolve convergently? And we need genomes to do that. And so I got excited when it occurred in 2009, 2010, as you heard from Harris, the Genome 10K Consortium formed by Ollie Ryder, Steve O'Brien, and David Hauser. And I got really excited, and I thought we can sequence all these bird genomes and the mammal genomes for the learners and the non-learners, and we can find the genes that are necessary for language and vocal learning. And they made me a bird chair. That was exciting. They introduced me to Goji Zhang and Tom Gilbert. The three of us together formed what we call the Avian Phylogenomics Consortium to sequence a bunch of bird genomes as part of this effort that BGI was supporting. Adam Philippi asked if he could help. I took a parrot genome, and we were applying what was called Roche 454 long reads at that time to see if we can get a high quality assembly. And Adam is the go-to person for high quality genomes. That parrot genome became part of an Assemblathon 2 competition for de novo assembly. And it generated some decent assemblies, but it was hard. And we weren't quite sure really the benefits of these higher quality or these short versus long read assemblies at that time. So what we ended up doing for this phylogenomics project is to sequence the genomes of 48 bird species that represent all bird orders, including vocal learners highlighted in red dots, and their closest relatives according to different phylogenetic trees highlighted in the white dots here. And with these genomes, we actually published over 50 papers, not all of them from my group, but I contributed to some of them. But these genomes really were able to advance science a lot, and we had a special issue in Science magazine on them. And one of the things I told you about we're asking is the evolution of the bird. Here is a tree that I had been using for 15 years prior to that. Here are the vocal learning bird species. And from this tree, based upon individual genes or whole genome alignments, not whole genome alignments, DNA hybridization, you can either propose 3 independent gains or a common ancestor and multiple losses. But remarkably, this brain pathways are quite similar. So lots of people were thinking that these 3 species, the vocal learning birds, basically have a common origin. So, or could the tree be wrong, basically? And so here is our genome scale tree from those 48 species, mostly with short reads, and the tree was wrong. Here are all the rearrangements we made in the species relationships. But the vocal learners were still far apart in the tree, particularly hummingbirds were much farther apart from parrots and songbirds, indicating, yes, that these brain pathways we think did evolve independently. And when we looked at individual genes and asked, are they regulated differently in the vocal learning brain regions of a songbird and a human, in light green here, compared to humans, to the surrounding brain regions? And the answer is yes. Work from Andreas Fenning and Greg Jedman in my lab, we found that the HVC song nucleus involved in production of vocalizations was similar to the human laryngeal motor cortex, particularly layers 2 and 3. The RA song nucleus of songbirds that sends the projections out of the brain to produce the learned sounds is also similar to the laryngeal motor cortex, to a different set of neurons. And in the striatum region of the brain in purple here, we found a similar region of the human brain with similar gene expression differences that's involved in speech production. We couldn't find these specialized changes in these genes in the non-vocal learning birds or non-human primates. Here's one example gene called SLIT1. It's an axon guidance ligand. It controls neural connectivity in the brain, and it is turned off. There's no white signal here of the mRNA in the brain regions that project out of the forebrain that control the ability to produce learned vocalizations. And you don't see that downregulation in the mRNA in the non-vocal learning birds like dove and quails. It's downregulated here in the human speech cortical regions. Actually, there are 2 of them. And these are the exact regions when surgeons put electrodes in the human brain during surgery that have the highest firing rates in red here when producing speech. So that we were really excited with these 48 bird genomes. And we now wanted to functionally characterize how are these genes developing these vocal learning circuits, these language-like circuits. And we started to dig it down into the individual mechanisms. And we discovered that these genes are highly fragmented in these assemblies. They have lots of gaps here between these different contigs and these light and dark brown colors. You can see the sequence quality drops down in these gaps. And we find this is not just the only gene. We find from many genes they were missing, or parts of the genes were missing. They were incorrectly assembled. Biological meaningful repetitive regions were misassembled or not assembled at all. This is causing many months to clone individual genes to correct them and so on. And many of us were working with artifactual gene structure and sequence and unknowingly making false conclusions about the biology. So at that juncture, the leaders of the G10K consortium asked, Eric, can you help us out and get more involved in the leadership of the G10K and do the same thing we did for birds now for the Genome 10K project? And I thought, great. And I started collaborating with Adam again. And we thought, you know what? We can't do this again. We need to do these genomes over again. And we need to figure out a method that's going to generate what we were calling platinum quality genomes. And so that's the second part of my talk before we start sequencing many species. And so we took one of those 48 birds, a hummingbird, because Jonas Korlak from PacBio volunteered to do it over again for us, with PacBio data. And we applied all the different available technologies at the time and in the subsequent years to this one species to test out different assembly approaches. And in the process of doing this, we set an aspired metric that we wanted to achieve. Have contiguous gapless sequence that's 1 million base pairs or longer in N50 contig values, scaffolds that are 10 million base pairs or longer, more than 90% of the sequence assigned to chromosomes, have the nucleotide base call accuracy to be above 40, which translates into no more than 1 error every 10,000 base pairs, and to have the maternal and paternal haplotypes separate out as much as possible. And you'll see the reasons why a little later. And a lot of this effort for testing out these various different assembly approaches was led by Ron Ray and Adam Phillippe's group and Shane McCarthy and Richard Durbin's group. And the formula that we came up with for the first phase of our project is to take PacBio long reads, make contigs out of them, link them up with 10x linked molecules, further link them up together in scaffolds with bionano optical maps, and then Hi-C linking up as well to get chromosomes. Fill in the gaps, polish up any assembly errors or let's say sequence errors in the long reads with the short reads, and then curate them, actually manually evaluate the assemblies, make corrections, and then assess what the assembly is like. And here is shown in a graph of the different data types and their length along the chromosomes, with the Hi-C being the longest length and the short reads, the long reads, even though they're long, being the shorter length. And the various different species that we actually tested out, 16 of them, and the people who have been sponsoring those genomes. This is a grassroots effort where we brought in lots of different people together to do this project. And one of the first analyses we've done, this is now kind of old news for some, but really it's important. What we found is that we could not generate contiguity to our metric unless we use long reads only. Here's the N50 contig values, are many-fold greater in contiguity with gapless sequence with long reads. Further, even though we can assemble genomes with short reads, they had tens of thousands of gaps, up to 70,000 here, compared to several hundred to several thousand with the long read-based assemblies. And further, in these gaps, there were thousands, 5,000 to 7,000 misjoins, contigs that don't belong together, that look like artificial rearrangements with these short read assemblies. What about the chromosomes themselves? Using Hi-C, Kirsten Howe's group, who led the curation team, basically will fix these assemblies and get what we consider in each one of these squares a chromosome. That's the Hi-C data mapped back to these scaffolds. And Marlies Cook then, for the hummingbird, has a karyotype map of the chromosomes of the hummingbird. And Martin Pippel from the Max Planck group developed a code that will take these Hi-C scaffolds, take the chromosome sizes here estimated from the images, and we find nearly a one-to-one correlation in the assembled scaffolds with the karyotype maps of these chromosomes. So we thought we're doing pretty good. Many of these genomes still had gaps in them, but some had just 1 or 0 gaps, particularly in the bird genomes, which have smaller sized genomes, where there is no problems here of collapse repeats or any problems that we can see. And we think they're telomere to telomere, not for the entire genome, but for some chromosomes. What about comparing to the old assemblies that were at chromosome level? Work of Joanna Damas and Harris Lewin's group found that if we take the new VGP assemblies and map them back to the old one, like the zebrafinch reference that thousands of scientists have been using in the past, we find that 2 chromosomes that were thought to be 2 different chromosomes are really 1 chromosome in our new assembly. Others called linkage groups we can now identify as chromosomes. Here you see these white open spaces here at the ends and sometimes in the middle of the chromosomes are newly assembled sequences added to the old chromosomes. And we also identify 7 new chromosomes that were previously not identified. What about a platypus, a sequence with a Sanger approach in the past? Here we find that we're adding more chromosomes. Well, we're adding, but there are lots of gaps in the old chromosome assembly here. And again, we also get chromosomes that belong to different pieces in the old assembly that really should be 2 separate pieces, that is, in the new assembly. Newly identified chromosomes, 8 of them here in the platypus. And also we have now, for the first time, identified the sex chromosomes. What about assembly that didn't use FISH or karyotyping mapping? Well, for the hummingbird, we have, looking at 300 kb or greater scaffolds, we have hundreds of them that are basically fragmented in the old assembly, now put together in full chromosomes in the new assembly, and thousands in this climbing perch fish genome here. So, and once again, the sex chromosomes identified. What about the annotations? A work of Françoise Theobald-Nissen from NCBI, taking the transcriptome data we had of these 3 different species here. We find there are many more transcripts, 5,000 to 20,000 more transcripts are identified, given all other things being equal. And when we look at those transcripts and ask, are they full length? We find now, compared to the new assemblies, roughly from 1,000 to 5,000 of the transcripts annotated in the old assemblies are incomplete. And we only have several hundred that are incomplete in the new assemblies when we look at the transcriptome data. And what is incomplete? So we started to look at across, is the incompleteness uniform across the genome, or is it biased? And we think it's biased. So work from Juwon Kim and Chul Lee from Seoul National University found that when they look at 100 base pair segments, the closer you get to a protein coding sequence here in the dark gray, the more missing sequence we have in these old Sanger-based or Illumina-based assemblies, such that when you get to the start site, anywhere from 30 to 50% of the genes could be missing the 100 base pairs in front of the first exon, and even the first exon. And then as you go to the exons and introns, you'll see differences in missing content here. And they're correlated with the GC content. So the more GC content you have, the more likely you're going to have it missing in your old genome assemblies. And that prompted us to look across the genome at protein coding sequences in general. And we see when you get to 1.5 kb within the start site of all the genes, 70% of the genes roughly in the genome, the GC content goes up. And then it starts to go down from the first exon and finally to get to the 3' UTR, and then goes back down to the level of the rest of the genome. And the introns, interestingly, have GC content that's similar to the rest of the genome. And there is differences between vertebrates, where birds and mammals have the highest GC content in their promoter regions and their protein coding sequences. Fish and sharks, a certain kind of fish, have intermediate levels, and bony fish, that's the teleus, actually have a drop in GC content. So we think they lost it, followed by this increase before they get to the exons here. So this has consequences in understanding gene regulation, because we find now, getting back to my questions about brain function, there's certain genes in the vertebrate brain, like the dopamine receptors that are high in the striatum, and we want to know what's causing their high regulation. Well, guess what? We find that in this GC-rich region that was missing in the black bar here in the old zebrafinch assembly is exactly where we find the epigenetic ATAC-seq open chromatin signal that we think is driving the expression of this gene in the striatum, but not the archopaleum. It's like a layer 5 equivalent of neurons in the bird brain for the cortex. Here, another gene, transcription factor upregulated in the archopaleum. What we find here, it's a multi-exon gene here. It's average GC content across the gene here in blue, but right in front of the promoter, here is this GC-rich GC content promoter region. It's missing in the old zebrafinch assembly. And once again, now the archopaleum has an open chromatin signal here as opposed to the striatum. We think could be driving the expression of the gene in this cell type. What about the vocal learning circuits? Here is a gene that is upregulated and specialized in the vocal learning nucleus area X. Once again, it also has the GC-rich region that's missing in the old assembly. The striatum, where area X is embedded, does not have a signal in the ATAC peak. The archopaleum, where it has high level, does have an ATAC peak. But area X, the specialized song learning nucleus, does have an open chromatin signal here, right in this GC-rich region, whereas the surrounding brain regions doesn't. And getting back to that SLIT1 gene I mentioned to you earlier, it also has this GC-rich region that's missing in the old assembly. So we're seeing this over and over again. The archopaleum, where it's upregulated, has a high signal, but we didn't see specialization in the RA song nucleus. So we think something else could be driving the down regulation here. But that GC-rich region bleeds into the first exon. And guess what? It's one of the missing exons we were struggling with to clone a few years earlier. And we find with the new assembly, actually the previous annotation predicted the wrong protein coding sequence because of this missing GC-rich region. Here is the true protein coding sequence now at the bottom, and even some other exons in some other GC-rich regions were also incorrectly annotated as a result of this missing sequence. SLIT1 is also downregulated by another gene called, is regulated by another gene called NeuroD6, a transcription factor. And so we began to look at that gene because it was also downregulated in the speech areas and the song areas of birds and in humans here in the speech motor cortex. And so Lindsay in the lab profiled the ATAC signal also for this gene. And we find not in the promoter region, not in that GC-rich region, but further out from the gene, 1,000 or so base pairs upstream, we find an ATAC-seq peak signal difference, high in the surrounding archopaleum, low in the the other way around, high in RA, low in the surrounding archopaleum. And this region, compared to in the genome of all other bird species, only the vocal learners have a mutation here in red, whereas all the other bird species we sequence do not have a mutation. They're highly conserved. And these mutations in the vocal learning species converts this region here into a androgen receptor transcription factor binding site. And so an androgen receptor, we know, influences the development of these vocal learning circuits in these birds. And so what do we think is going on here? We think that vocal learning birds evolve novel mutations here in blue that allows this receptor to bind. and we think to turn off NeuroD6, which then turns off the expression of the SLP1 gene in the vocal learning circuits, which will then allow certain connections to form. What about, how do you do that only in the vocal learning circuits and not in the other brain regions? Well, we think what happens here, histones come in and they bind to this novel sequence here and prevent the receptor from binding there, but it can bind here to regulate it only in the speech areas or the vocal learning areas in these birds. So that's a case study of what happens for regulation. What about, I said, the haplotype problem we were having? Well, here is a gene that's specialized in its upregulation by singing behavior in these vocal learning circuits in the vocal learning birds, but not the surrounding motor areas when the bird is hopping. Well, we find that this region has a highly repetitive sequence upstream of its promoter region. And when we found we're assembling these long read-based assemblies, we discovered that they were accidentally initially put together as repetitive sequence one after another, where they really belong to two separate haplotypes. And we think that only the vocal learners have these repetitive sequences. And so we're getting screwed up assemblies only in the vocal learning species because of this repetitive DNA here. And this led us to look further in other studies into the causes of these false, what we call now haplotype duplications. And here's a model from Erich Jarvis, where naturally in the genome you have maternal and paternal haplotypes. And some parts of the genome, the mother and the father DNA basically is more divergent than the rest of the genome. And the assemblers then treat this as two different genes as opposed to one gene. It forms this bubble in the assembly in this divergent gene. And then they either get pulled apart here, or one of them is entirely separated from the other. And then when you come and scaffold these contigs together, the assembly tool then puts a gap between these two different haplotypes and treats them as two separate genes, as sister genes, as opposed to homologous genes, either in the same chromosome or in different chromosomes. And when now we have algorithms, including some developed by the VGP group called Purge Dupes from Richard Durbin's group, that identifies these errors and tries to actually pull them apart so that you don't have these mistakes of pseudo-duplicated genes. But here we found many cases of them in the older assemblies, where in the new VGP zebrafinch assembly, work of Byung-Jun Ko has found that some of the older assemblies have two entirely duplicated genes that should not be, or some exons that are duplicated, or even just one individual intron that's duplicated and put into one gene as opposed to the other, causing false whole gene duplications or partial gene duplications. And what about going beyond individual genes? The work of Konstantina Theofanopoulos with my group has looked at synteny across all these species, particularly for the vasotosin receptor family, and has found in some of these old assemblies, like for the platypus, they're scattered across multiple scaffolds, looking at the synteny with many different gaps. In the new assemblies, we find all the genes are expanded because we're getting more gene sequence. Further, this region is in one entire contig here, gapless. And now with that, we can infer the true evolution of this gene family from the common ancestor from vertebrates, initial gene duplication followed by a whole genome duplication, creating two extra copies of this gene family, including the oxytocin receptor, followed by further segmental duplications and deletions, and getting rid of all false duplications so we don't incorrectly infer a gene duplication that was really an assembly error. Now we can get better accuracy of genome assemblies, genome evolution. But yet, we still have some problems to solve, particularly repeats. Looking at repeats, basically, the more repeat content we find in the genome, the poorer the contiguity here, the more gaps we have in the assembly, the number of collapses we see of repeats piling up on top of each other, and the greater false duplications we see here. Also, the greater the heterozygosity, the more false duplications we have. These purging tools I mentioned before, they get rid of a bunch of these false duplications here, including those that are involving repeats, but we still got some more improvements to go. One of the most promising approaches is around, developed by Sergey Karen and Eran Ray and Adam's group, takes a parental DNA, in this case showing for an example zebrafinch, the dad and the mother DNA of short reads, sorts out the haplotype long reads of the child, and then assembles them independently. And we can get highly contiguous NG50s here for both the maternal and paternal haplotype in what we call these trio-based assemblies. And without purging, we can find that we go from 5% false duplications down to 1.4 to 1.3%, doing much better here to separate out these haplotypes and prevent these false duplications. So I'll end here on what we are now, some of us are calling platinum quality genomes. There's more work to go, but we're doing much better. I can answer my vocal learning questions now with these genomes. So what do we do for these large scale projects? So we decided for the goal of the BGP is not just to sequence 10,000 vertebrate species for the Genome 10K effort, but all 70,000 species, and to generate them at least at high quality, error-free, near gapless, chromosomal level, haplotype phase, annotated as well, reference for all extant vertebrate species, and utilize those genomes to address fundamental questions in biology, disease, and conservation, as you heard from Harris, and to contribute these genomes to other large-scale projects like the EVP, the Darwin Tree of Life, and collaborate with them. And we like to do this in phases. First is in the phylogenetic scale here, all orders, all families, then all genera, and all species, where each phase here reaches a certain milestone of various different projects like EVP families or the G10K milestones or the B10K, BAT1K, and so forth, for groups you'll hear from in this conference. And for this first phase, we decided to use divergence time to separate out species according to orders, where we take all species that have a common ancestor that goes back 50 million years or more, and shown here for birds and mammals, which adds up to 260 species. You saw this early in Harris's talk. And here is the breakdown of the various different vertebrate lineages. And to make this work over the last 4 years since we started this, we really had to build up a sample collection and coordination team, write new kinds of permits to do things at scale. We had to form data production groups, assembly algorithms, and curation teams, and annotation improvements, and websites, and databases, and annual meetings like this one here, and do outreach to the community. And also have the consortium not just generate the genomes and build the infrastructure, but also bring in the people who are doing the biological analysis with these genomes. And so here is the Vertebrate Genomes Project website. I encourage you to go to it. If you click here on what's called the Genome Arc, you'll see here all the data that we're depositing for the various species here. We now have 139 species in there, and 92 of them have highly curated. We're also depositing the data in the NCBI database here, and all assemblies in this aspect of the database reach the VGP metric. There are 102 species there now that are either now available, annotated, or in progress, and more to go. And also the UCSC Genome Browser Group has taken these genomes and created browsers of them. And I encourage you to go use them there for those who like to use this tool. The Ensemble Group has also done something similar and have a VGP portal for these high quality reference genomes and to go use them there. And we're partnering with the B10K consortium for at least some of these species to produce high quality bird genomes. The BAT1K group, you'll hear from Emma Teeling and Sonia Vernes later on. And for B10K, you'll hear from Goji and Stephanie Tilner. And you heard from Harris about the Earth Biogenome Project already. And so we'll satisfy the family level assemblies at least in terms of reaching that metric for that group. And other groups have been forming, and I want to alert you to a new one called the European Reference Genome Atlas, whose mission it is to sequence the genomes of all 200 eukaryotic species in Europe. And leading that charge is the Darwin Tree of Life project. You'll hear from also later in this conference. And they are contributing some genomes to this first phase of the BGP project. And I thank them for that. And the tools that we've been developing and the lessons learned for the BGP project has not only influenced the other projects that I just mentioned, but also a new pan-human reference genome project that I and others are part of as part of this consortium, where the goal here is to generate not just a new human reference reaching this quality, even telomere to telomere for all chromosomes, but also to actually generate a reference that represents human diversity, from 350 or more people around the planet, representing all different genotypes, using high quality approaches like we've been developing for the VGP. And now we're all learning from each other. I encourage you to go look at the website there. So future plans for the VGP. We'd like to complete phase 1 by 2021. We have 120 species done. We have 140 more to go. We have all the funding we need for that 140. Now we just need to do it. We need to conduct foundational studies on all vertebrate orders with those 260. We want to work on what we're calling a VGP version 2 pipeline, testing new technology that has been developed since, testing Hi-Fi from PacBio, ultra long nanopore reads from BioNano, Hi-C from various companies, BioNano, trios, and non-trios. And the Hi-C we've been using is from Genomics and Dovetail Genomics. We still need to set annotation and alignment standards. We still need funding for phase 2 for 1,200 vertebrate families. Our eventual goal is to develop a high throughput approach that can give us T2T diploid assemblies. And we want to eventually get to the point, as Gene Myers says, to do a genome only once, so we don't have to keep doing it over and over and over again, and then get to the point where we can then think about the next stage of genomics. And so I want to end here giving thanks, particularly to the Assembly Working Group, a group that's been hard at work for the last 3 to 4 years to get us where we are at now, particularly from Adam Phillippe's group at NIH and Richard Durbin's group at the Sanger and University of Cambridge, Gene Myers' group at Max Planck, who've been instrumental in these assembly approaches that we've been developing. Harris, Lewin, and Joanna, you heard from my group with Olivier Federigo at the Rockefeller University, Kirsten Howe's group leading the curation team at Sanger, Francoise and Fergal Martin at the NCBI and EBI for the annotations, Hebao Kim's group at Seoul National University, and Goji Zheng's group at BGI, and others like Simone and Mark who have joined us from other places in the various consortiums. And to thank the VGP consortium members that contributed to our first paper that's submitted on bioarchives and that's currently under review, which we hope will come out soon, and it lists all the authors here. The companies that have helped us from PacBio, BioNano, Genomics, I've named them throughout the talk, as well as the infrastructure support we got from the various funders like Howard Hughes, Rockefeller, Sanger, University of Cambridge, Max Planck, and NIH, and the grassroots efforts of many scientists who have contributed funding upwards of $60 million in the last 4 years to contribute to these 260 phase 1 species. And finally, the conference organizing team, who you've heard from Mark, Blaxter, Harris, myself, Victor Lopez, and Sophie Potter, and Chloe Leach, and Nicolette Capriello, and Sadie Paez for their support. And I will go ahead and stop here and take questions, and thank you for your attention.
Really appreciate it. Lots of great questions have come in. And so I'll start it off from an anonymous question. How much do these platinum level long read based genomes cost? And at which steps are improvements shortcuts most likely to lead to cost reductions?
Yes, so as you saw, we had a combination of 4 technologies in our first pipeline. And because of that, we have to pay a lot for different reagents and so forth, and the labor and effort. So just a 1 gigabase genome, initially it was costing us somewhere between $15,000 to $20,000 overall amongst the various groups. Now we've got it down to $6,000. And this includes, discounts we get and so forth. So that's a big reduction, and we think we're going to get it down to half that price in the coming year or so. So to put it into perspective, these are chromosomal level genome assemblies that are almost as good as, if not as good as, the initial human genome, which cost $3 billion or more. So 15,000 down to 6,000 is not so bad. Well, for one genome, yes. And where is the next bottleneck? The next bottleneck, I think, is really in the algorithms more than the data types. I think the data quality is now better than the algorithms are used to. And so we need to improve upon the algorithms, and then, as always, get the reads to be longer and get them to be more accurate.
Okay, thanks. Another question about assemblies. Interesting question. How to generate platinum quality sequences from degraded DNA of poorly preserved museum specimens?
That is hard because some of a lot of that degraded DNA is going to be shorter than the read lengths than the technology can handle today. So with PacBio and nanopore reads, you can get from 10, 20, 100 KB reads, or even more in some cases. Whereas the reads in the degraded DNA might be 100 base pairs, 200, or down to 30 base pairs. So that I think is going to require algorithms that one day make short reads assemble to the high quality you get in long reads. And so far we have not seen that yet. So I think it's going to improve in the coming years for partially degraded DNA, but not by enough unless something really new happens with short reads.
Right, but we'll take them if that's the only thing we have.
If that's
right?
the only thing available, a museum specimen of an extinct animal with no fresh DNA, we'll take it. Yeah.
Indeed. Okay, another question. How do you annotate new genomes? Which pipeline and software?
Yes, this has been a challenge to scale up on the annotations, but two, to how, what standards do you use and when do you stop? And one thing I learned about annotations compared to genomes is that genomes, you can see the end in sight. You know what the end is, an error-free, complete telomere to telomere assembly. Annotations is a bottomless pit because genes could be regulated. You can find all these different regulatory regions depending on the tissue type, the age of the person, and so forth. So where we are at now, according to Francoise and Fergal, is basically to get a decent annotation, for, well, let's say 80, 90% of the genes, you think, you need about 5 different tissues. A combination of short reads and long reads would help because you get splice variants better assembled with the long reads. And you need diverse tissues. And what we've been focusing on is the brain has the greatest gene diversity. The liver also has gene diversity, and the gonads.
Okay.
Oh, and the algorithms, I don't know yet. We're going to hear from the annotation groups, but the Sanger team or the EBI team and NCBI are working on algorithms to make more rapid annotations doable.
Yeah, so the listener should tune in to the annotation section chaired by Paul Fleesek. Okay, and lots of questions on assembly, Eric.
Go ahead, I'll try to swing them quickly.
The next one is, how applicable are these new sequencing methods to field-based studies, which cannot flash freeze their specimens, but rely on ethanol and other?
Yeah, so the VGP sample preparation group and sample preparation groups of other consortiums have been working on this problem exactly for that question. And we find that it is actually possible to store samples, in small volumes, in small volume of the sample itself, in alcohol, in ethanol, at room temperature for a day or two before you actually freeze it to get high quality DNA. It's not the preferred method, but it is possible. You don't want it to go out 1 week, but it can be for a few days. And we have a study being prepared on that too that will discuss that.
Great. Now, is the lack of GC regions a sequencing Illumina problem or an assembly artifact?
It's a sequencing problem. So what it is that the enzymes for Sanger-based sequencing and the enzymes for Illumina-based sequencing have difficulty getting through GC-rich regions because they fold up and so forth. There's a lot of secondary structure there. Whereas in some of the single molecule methods, they go through a pore, or they have methods that unfold the DNA better. Also, some of the enzymes can get through them better. And so they're just not in the raw reads to begin with in many cases.
Great. Another question. I think we have time. Good question here. partially addressed it, and I think it's very important to consider this. What is the utility of these new long-read-based genomes for studying complex genomic regions like the MHC?
Yes, we think the utility is essential for these high-quality genomes because genes like the MHC, which have a lot of redundancy and thus duplication, making it more difficult to assemble. So these are genes involved in immune response, for those who don't know. We're finding they're better assembled. We don't actually have them perfect yet, but they're much better assembled. And we can learn about immunity, including for COVID-19 infections, because they're not just the only genes that have a lot of redundancy in the immune system. We can learn about immunity better from these higher quality assemblies and what we once thought were correct assemblies or not, once we see these new assemblies with these more redundant genes.
Great. Well, Eric, I think I'll close it there, and I'll close it with the final, one of the questions, which was actually a comment, which was liked by 19 people. And that is totally brilliant. So thank you very much, Eric.
Well, and I want to thank that brilliance is to really thank those consortium members I showed at the end. So I'm just the representative, the messenger here. And this really takes a village to make a project like this work, and not just a village in one particular university or country, but around the world.
Indeed. Yeah. So true. Thank you very much, Eric. And we'll be back shortly with the beginning of parallel sessions this afternoon. So please come back in about 10 minutes time and we shall begin then.